Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is transporting plant. In the previous class, we have already talked about some of the introductory part, and along with that, we have also discussed about the different means or different types of transport. Okay, now in today's lecture, we are going to deal with mainly short distance transport and their mechanism, okay, and how the plant show a relation with the water that we are going to discuss. So we are going to discuss plant water relations. And when we talk about the plant and water, we know that the plant is mainly made up of water. It has a too much amount of water. And in different plant, the content of this water is variable. So if we talk about water, it is obviously essential or it is required for all physiological activities of the plant. Whatever be the physiological, there is a photosynthesis, respiration, growth, development. For all this, the water is required, and it plays an important role in the living organism. Obviously, it provides the medium in which most substances are dissolved. Obviously, majority substances are get dissolved in this one. And when we talk about a herbaceous plant, we know that herbaceous plants have only 10 to 15% of their fresh weight as dry matter. Means if we take a fresh weight of herbaceous plant, only 10 to 15% you will see the dry matter. Otherwise, other remaining part will be the water. Water means most herbaceous plant have 85 to 90% water. Okay, these are the herbaceous plants. Now, when we talk about another example, that is a watermelon. That watermelon contains over 92% water. When we eat a watermelon, it is rich in water, so it has a 92% water. So this is the watermelon, as we know. Now the distribution of water within a plant varies because woody parts have a relatively very little water, while the soft part contain mostly water. So this is the hard part have less water and soft part has a high content of water. When we talk about a seed, in seed also you will see that they may appear dry, but it still has water. Otherwise, it would not be alive and respiring. Without water, they cannot respire, they cannot alive, and if they cannot respire, they will not germinate. So they have still some content of water. Okay, in case of the seed. When we talk about a terrestrial plant, they take up a huge amount of water daily, and in that, most of it is lost to the air through the evaporation from the leaves, that is the transpiration. Okay, so terrestrial plant take up a huge amount of water daily, they absorb too much amount of water, but majority of that getting lost through the transpiration from their particular surfaces. Now in that, if we take an example, the mature corn plant that absorbed almost 3 liters of water a day. If we take a mature corn plant or a maize, it will absorb 3 liters of a day of water a day. And when we talk about the mustard, this is a particular mustard. In mustard, you will see that the mustard plant absorb water equal to its own weight in about 5 hours. In about 5 hours, it will absorb water equivalent to their own weight of the plant, that is mustard. So this much water need and water content is found in the different plants. Because of this, we tell that water is essential for the all physiological activities of the plant. And obviously, water is a very good solvent as well. And as we have discussed, they provide a medium in which different molecules are dissolved and suspended. So this is the what water content. Now water is present within the cell, and when we talk about a plant cell, they have a cell wall and cell membrane. In animal cell, the outermost will be the cell membrane. So because of high demand of water, it is not surprising that water is often a limiting factor for plants growth and productivity in both agriculture and natural environment. Okay, due to this demand, it is obviously acting as a limiting factor for the plant growth and productivity in agriculture and environmental ecosystem. Now, When we talk about a plant, plant cell have a cell membrane, and when we talk about the membrane permeability, so now we are going to talk about the different membrane permeability, and on the basis of permeability, we are going to talk about types of membrane. So we are going to discuss types of membrane based on permeability. Based on permeability. Okay, and in that the first one is permeable membrane. If we talk about permeable membrane and if we define this permeable membrane, then this membrane allows the transport of both solvent and solute. This membrane allows transport of both solute and solvent. Okay, it allows a transport of both solutes and solvent across it. And if we take an example of this permeable membrane, then it is a cellulosic cell wall, or we can say primary cell wall. Whereas the second type of membrane, which are known as semi-permeable membrane, and semi-permeable membrane is a membrane this membrane allows transport of this membrane allows transport of solvent only, but not solutes, but not of solute across it, and that are known as semi-permeable membranes, so which allow the transport of solvent only. And if example we take of a semi-permeable membrane, that is a animal urinary bladder, cellophane, parchment, paper, and copper ferricyanide membrane. These all are the examples of the semi-permeable membrane which allow the transport of solvent only. Now after this, the third type of membrane are selectively or differentially permeable membrane. Selectively or differentially per membrane. And selectively or differentially permeable membrane, if we talk, these membranes allow transport of this membrane allow transport of solvent and along with it selected solutes. And if we talk about the examples of selectively or differentially permeable membrane, then the example is cell membrane or plasma membrane, then tonoplast, then organelle membrane, that all are selectively or differentially permeable membrane. And the next one is impermeable membrane. If we talk about the impermeable membrane, this membrane neither allows solute allow solute nor solvent across it. So it is not going to allow a solute as well as solvent. Then it is known as impermeable membrane. And if we talk about the examples of a impermeable membrane, that is suberized cell wall, that is cork, then cutinized cell wall, that is epidermis, then polythene, glass, plastic, etc. These all are impermeable membrane. Okay, so there is a permeable membrane, semi-permeable membrane, selectively permeable membrane, and impermeable membrane. These all types of membrane can be there in a plant. Now, after this, if we talk the particularly the after studying this membrane, if we talk about the different types of conditions or different types of mechanism which are involved in the transport of molecules in the plant, then first we are going to discuss in that is osmosis. Now, when we talk about osmosis, the plant cell surrounded by a cell membrane and cell wall, that we know very well. Okay. Now, when there is a cell membrane and cell wall around the plant cell, then the cell wall is free permeable to water and substances in solution, but cell membrane is selectively permeable. Now, hence cell wall is not a barrier to the movement. The barrier to the movement will act as nothing but a cell membrane. Okay. So, in a plant cells, the cell membrane and the membrane of vacuole, that is tonoplast, together are important for the movement of molecules in and out of the cell, because both are selectively permeable membrane, the cell membrane as well as what we can say the tonoplast. So if they are selectively permeable membrane, they allow the molecules transport selectively through them. Now when we talk about osmosis, osmosis is the term that used to refer the specially to the diffusion of water across a semi-permeable membrane. Okay, so it is osmosis going to talk about a transport of solvent, permeability of solvent, diffusion of solvent, that is a water, that is through the semi-permeable membrane. Then we call it as osmosis. Osmosis occurs spontaneously in response to the driving force. It occurs spontaneously, and when there is a particularly this particular solution in equilibrium, the molecules will transport from higher concentration to lower concentration through the semi-permeable, and these molecules are of water. So when water molecules move or solvent move from that higher concentration to lower concentration through the semi-permeable membrane, that is defined as osmosis. And in this, the net direction and rate of osmosis depends on, okay, that we will discuss. Before that, we have to discuss one more point. So if we talk about the definition, they don't can have only one definition. So first we remember that the term osmosis coined by Nollet. Okay, and here If we define osmosis, it can be defined as transport or diffusion of solvent through semi-permeable membrane from higher concentration of solvent to lower concentration of solvent, or we can define it as from dilute solution to concentrated solution, or from less concentrated solution to highly concentrated solution. So all higher concentration of solvent can be present in dilute solution and less concentrated solution as well. Because of this, we are going that they can move from a dilute or less concentrated solution to the concentrated solution. Then also it can be referred as a particular osmosis. Now obviously it is a type of diffusion. Okay. Now the net direction and the rate of osmosis. It is also known as that the rate of So net direction and rate of osmosis depends on pressure gradient. Obviously, if there is a high pressure to lower pressure, the movement will of molecule will take place of water. Then also the concentration gradient. If the concentration is solvent is higher and one side is a low concentration of solvent is there, if the difference is more, the gradient is more, then the direction will occur according to the gradient. And it is also known that most of water is absorbed by the plants through the osmosis made that occurs due to the concentration gradient and does not require any metabolic energy. Okay, so it is a passive absorption. Okay, and in passive absorption, most of water is absorbed by the plant through osmosis. Okay, and that is due to the concentration gradient. And obviously, it is a passive absorption. It is a passive type of absorption, so energy is not required because it is from higher concentration to lower concentration. Now, water when moves from its region of higher concentration or higher chemical potential to the region of lower chemical potential or low concentration, and this movement will continue till the equilibrium is reached. And at equilibrium, the both the side or both the chambers will have the same water potential, or same we can say the water content or the solvent content. Okay, so that is particularly we can say the osmosis. Now, when in osmosis, a molecules move from higher concentration to lower concentration, that is water molecule. To explain that how the osmosis takes place, there is a two experiment that we will discuss: potato osmometer and thistle funnel experiment. In potato osmometer experiment, it was done that if a potato tuber containing a concentrated solution of sugar is placed in a beaker, here what is we'll do? We'll take a concentrated solution sugar in the potato tuber by cutting a potato tuber in a proper way and placing them in a beaker which containing a normal water. Now, while placing, for taking a sugar solution in the potato, a cavity is made which has a sugar solution and it collect the water from the surrounding beaker due to osmosis. So see, this is the diagram. Okay, this both are the potato, and in center you can see the cavity is done, and this both have a sugar solution, and both beaker have water. Before osmosis, you can see that the sugar solution concentration is this much. Okay, and when osmosis takes place, the water enters, and after entering the water, the sugar solution level get increased, And when sugar solution level is getting increased, okay, it is after osmosis. Okay, so before osmosis and after osmosis, the sugar solution level is going to be increased here. Now these are the two beaker A and B. Suppose let us study this figure, okay, in which two chambers A and B containing solution separated by a semi-permeable membrane. You can see this is a semi-permeable membrane. Here it is NCRT diagram. It is a solute molecules. Okay, this is the water, and here less solute molecules are there, and here high solute molecules are there. Now the question can be asked for the what particularly this, and the question can be asked that the solution of which chemical has a lower water potential. Okay, so this is the question of water potential. So we will discuss this question in a later part when we will discuss the water potential actually. Okay, but you should remember this diagram. This is an important one, and we have to discuss this question in a later part. Okay, we will discuss that. Now let's discuss the another experiment, that is a Thistle funnel experiment. This experiment also used to demonstrate the osmosis. It is also demonstrating osmosis. And in this experiment, a solution of a sucrose is taken in a Thistle funnel. Solution of a sucrose is taken in a Thistle funnel, and which is separated from the pure water in a beaker through the semi-permeable membrane. Okay, so this is the beaker. They have a sugar solution or sucrose solution. Okay, and this is the water, and the water is there. Now water intake will occur. So in this experiment, a solution of sucrose is taken, okay, in a Thistle funnel, okay, and which is separated from the pure water in a beaker. Now when osmosis will take place, this is a semi-permeable membrane. So when osmosis will take place, water will enter from here to here, and this sugar solution, which was this much, that get increased up to this level, and increase in this sugar solution. Here you can see this. Suppose it was initially it was up to this level, but now it has increased up to this level. Means that this is showing that osmosis has occurred. But when we want to stop this entry of water here, we have to apply a pressure from this top side to prevent the entry of water in this beaker from this Thistle funnel. And if you want to block that, we have to apply a pressure from this side, top side. Then the entry will be stopped, and that pressure will be known as osmotic pressure. So water will move from a funnel into the funnel, resulting in increasing the level of solution in the funnel, and that is showing osmosis is occurring. And this will continue till the equilibrium is. The equilibrium in the beaker and the Thistle funnel will accomplish till that this is going to take place. Now to prevent the entry, external pressure is applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane, and that pressure is known as osmotic pressure. And this pressure is required to prevent the entry of water or water from diffusing in. In fact, the osmotic pressure, and this is the function of the solute concentration. Okay, so to prevent the entry of water, so if we talk about the cell, when in a cell the entry of water stops, means we can say that osmotic pressure has reached. Okay, if the concentration of solute is there more in the cell, the more water entry will occur and more osmotic pressure will be required. Okay, so same way, if solute concentration is more in this beaker or in this Thistle funnel, then more pressure will be required to prevent the entry of water. So osmotic pressure is directly proportional to the concentration of solute in a solution. If a solution has more solute concentration, the osmotic pressure will be high. Okay, so the more the solute, the greater will be the pressure required to prevent the water from diffusing in. Numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. The sign of osmotic potential, or we can say the solute potential, is different. And solute potential here, if we talk osmotic pressure, this osmotic pressure is represented by pi, and this osmotic potential is known as solute potential. This potential is known as solute potential, and osmotic pressure is equivalent to the osmotic potential, but sign is opposite. Is osmotic potential or solute potential is known as psi s, and it is negative where it is a positive. So the osmotic pressure is positive pressure applied while osmotic potential is negative. So if we compare that osmotic potential, solid potential is what? A potential of solute to lower the water potential. Means if more solute is there, more negative solid potential will be there. So if we talk about the solute potential, if the osmotic pressure is a 5 atmospheric pressure, then the solute potential becomes -5. So solute potential is equal to minus of pi a, pi. That you should remember. That is the comparison. Now, to understand this osmotic pressure, after understanding these two different things, we have to understand the osmotic pressure in more detail. By after discussing this osmotic, we can say thistle funnel experiment and potato osmometer, that is osmotic pressure. And this osmotic pressure is represented by pi. The term was coined by coined by Pfeffer. And it is a pressure developed in osmotically active solution when the solution is separated from the hypertonic solution. Okay, so if we define it, it is a pressure developed in an osmotically active solution, that is hypertonic solution, or we can say concentrated solution, when this solution is separated from hypotonic solution. Hypotonic solution, or its solvent by means of semi-permeable membrane. And this osmotic pressure is denoted by pi. Now, if we talk about a mathematical expression, then mathematical expression of osmotic pressure is explained by want of formula. And want of formula that represent osmotic pressure, that is pi, is equal to ICRT. That is the formula of osmotic pressure. Pi is equal to ICRT. And in this particular formula, I is equal to ionization constant, C is concentration, R is gas constant. Gas constant is 0.082 mole per molecule, and T is absolute temperature. So all these things are there. So if you want to calculate the osmotic pressure of 1 mole of glucose, then we can calculate by this formula. So the question is, calculate the value of osmotic pressure of 1 mole glucose solution at 0 degree Celsius temperature. And here the solution, if we talk, then that is pi is equal to 1 into CRT. So C is 1, gas constant is 0.082, and temperature is 273 Kelvin. So it is going to be 22.4 atmospheric pressure. Okay, that is osmotic pressure. Now the osmotic pressure is here is 22.4. Okay, but if we take calculate the osmotic pressure of glucose of NaCl, that is electrolyte, then question is calculate the osmotic pressure of 1 mole NaCl. Now if we talk about solution, here NaCl get dissociated into Na plus and Cl minus. So here the I, that is ionization constant, is become 2. So here pi is equal to ICRT. Here I is 2 and CRT will be 22.4. So it is going to be 44.8 atmospheric pressure. And this 44.8 atmospheric pressure is a particularly, we can say, the solution, the pressure. So NaCl have a more osmotic pressure and the glucose have a comparatively, we can say, a less osmotic pressure. Okay, so the molecules are, if we talk, there are more. So if the solute molecules are more in NaCl, and if we take an example where a glucose is separated from the NaCl, then the water will move from a low osmotic pressure to high osmotic pressure. That is from glucose to the, we can say, NaCl. So according to osmotic pressure, we can say water or solvent move from low osmotic pressure to high osmotic pressure. So low osmotic pressure to high osmotic pressure, the movement will be occurred, and that is what we can say the osmosis. Now, when we talk about the osmosis, then now we have understood that osmotic pressure of electrolytes is greater than non-electrolytes that we have discussed. And it is also known that osmotic pressure is variable in plants of different habitat. And if we talk about the ascending order of osmotic pressure, then the ascending order of osmotic pressure is there, and in that we can talk that hydrophytes have osmotic pressure lesser than the mesophytes. They have lesser than the xerophytes, and the lesser, that is the halophytes. So halophytes have a highest osmotic pressure or Highest osmotic pressure is seen in case of the halophytes, that is salty area plant, like mangroves. Now, in that, one more important point you should remember that highest osmotic pressure is reported in halophytic plant, in halophytic plant, that is Atriplex convertifolia. Okay, in that they have a highest osmotic pressure, that is 202 202.5 atmospheric pressure. Okay, then it is also known that osmotic pressure of pure water is taken to be zero. Then this osmotic pressure is measured by osmometer. Okay, so that is what particularly we can say the osmosis and all the things with respect to the osmosis. An experimental demonstration also we have understood, and what is osmotic pressure and how it will develop, that all thing we have discussed. Now, after osmosis, if we talk about the some questions, a watermelon contain blank amount of water, and we have discussed they have too much water, 92% of water it has. And for a solution at atmospheric pressure, water potential is equal to solute potential. Actually, this is a question of water potential. We will discuss that. And a mature corn plant absorb almost 92% of water, 3 liters of water in a day. Now, after osmosis, next we are going to discuss one more mechanism, an important mechanism, that is plasmolysis. And here we are going to talk about the behavior of plant cell or tissue with regard to the water movement depending on the surrounding solution. That is the behavior of plant cell in a hypertonic solution. We are mainly going to explain this, the plasmolysis. So here we are going to discuss the so it is a behavior of plant cell in hypertonic solution. Okay, so in that hypertonic solution, this behavior is shown by plasmolysis. So first we have to discuss the different type of solution. And in that first we are going to talk about the isotonic solution. The isotonic solution is a solution in which the external solution balances the osmotic pressure of the cytoplasm. Means both have a same osmotic pressure. Now, how it will be possible? If both have a same concentration. So here suppose this is the isotonic solution. This is the A system and this is the B system. Both are separated by semi-permeable membrane. This also have a 5% solute and 5% solute here also. 95% water and 95%. Means both are in isotonic condition. They have equal solute concentration, equal concentration of both the solution in this A and B. So because of this, they are in equilibrium, we can say, and they are isotonic solution. So now what will happen? Both are in equilibrium. So in when the outer solution is the balanced one, suppose this is a cell and I keep this cell in the particularly a beaker, and that beaker has isotonic solution, then the concentration of solution of the cell and this both have a same. So intake and outgo of water will take place. Okay, so intake and outward movement of water takes place, and both sides the movement is going to occur, but net movement becomes zero. Okay, the flow will not stop, that will occur in both direction, but the net flow will be not there. So it is known as isotonic solution. Now another one is the hypo. Hypo means low. Low concentration is there in the solution. External solution is more dilute or it is a less concentration than the cytoplasm. Then we call it the hypotonic solution. So in hypotonic solution, if we take a particular weak cell and place in a hypotonic solution, which is a more dilute than the cytoplasm, okay, and it is the particular, this is more concentrated, so water will move inside, and hydroponic condition cell get swollen, and this swollen cell is known as turgid cell. Whereas when there is an intake and outlet in equal direction in the isotonic solution, that type of cell are known as flaccid cells. The swollen cells are known as turgid cell, and they form turgid, they become turgid in the hypotonic condition. Whereas in hypertonic, the external solution is more concentrated than the cytoplasm, and if the external solution is more concentrated, the water will go out. So suppose this is a cell, and this is a hypertonic solution. Then water will move inside, and when water will move inside, this particularly, sorry, this is a hypertonic. So here what will happen? Water will go outside, and when water will go outside from this concentrated cell, less concentrated to high concentrated cell as solution, that is hypertonic, the shrunken cell will be obtained, and this shrunken cell will be known as plasmolyzed cell. Okay, so in hypotonic, the turgid cell will be formed. In hypertonic, the plasmolyzed cell will be formed. In isotonic, the cell will be called as a flaccid cell. So this all are the hypotonic solution. If an animal cell is placed in a hypotonic solution, it is going to burst. But the plant cell have a cell wall, so they cannot burst. Okay, but because in hypotonic water entry will take place. In isotonic, both intake and outtake is same in both, particularly animal and plant cell, and they are known as acid. But in hypertonic, the particular loss will of water take place, and they get shriveled or shrunken, and the shrunken cell are known as plasmolyzed cell in case of the plant. So cells swell in hypotonic solution and shrink in hypertonic ones. That is clear from this side. And the plasmolysis occur when water moves out of the cell, when a cell membrane or through a cell membrane of a plant cell in a hypertonic solution, and due to the shrinks away from its cell wall, the plasma membrane shrinks away from cell wall, that is known as plasmolysis. So when water go outside, okay, water go outside, then the shrinking will occur, and this shrinking is known as particularly, this loss of water and shrinking is known as plasmolysis. You can see this is the example of onion plasmolysis, and this occurs when a cell is placed in a solution that is hypertonic or has a more solute than the protoplasm. When water moves out of the cell, it is known as exosmosis, and it is lost from first from the cytoplasm to outside, and then from the vacuole. Okay, so when plasmolysis occur, the water loss, because outer side concentration is hypertonic, and exoosmosis will take place. First, the water will move out of the what, particularly cytoplasm, and then it will move out of the what, vacuole. Okay, so the water, when drawn out of the cell through the diffusion into the extracellular fluid, causes the protoplast to shrink away from the cell wall. And when protoplast shrink away from the cell wall, this is known as the shrunken cell will be obtained, and this shrunken cell will be known as a what we can say, the plasmolyzed cell. So initially, this phenomena is indicated by a shrinkage of the protoplast, leading to the separation of the plasma membrane from the cell wall. And in this, the separation of plasma membrane from the cell wall takes place. Okay, so hypotonic, isotonic, hypertonic, what will take place that we know. And in that, the first, it occurs in 3 steps. Okay, there is a limiting plasmolysis. Sorry, we can say limiting, yes, limiting plasmolysis, incipient plasmolysis, and the, we can say the evident plasmolysis. So this occur in 3 steps. So first we talk about steps of plasmolysis, and the first step is limiting plasmolysis. In limiting plasmolysis, the water wall will loss out. Suppose this is a cell, plant cell, this is cell wall, and this is a cell membrane. So when it is placed in a hypertonic condition, the water will loss start. And as the water move out, in this first, the cell membrane gets separated from the corners. So in limiting plasmolysis, cell membrane gets separated from corners. And when the cell membrane gets separated from the corners, here, suppose this is a flaccid cell which has a turgor pressure 0, then here it is a turgid cell and the water loss will take place. So here it has a turgor pressure positive, then in the limiting plasmolysis, the turgor pressure becomes 0 in the cell. And here in the first stage, sorry, this separation will take place in the later one. First, what happens in see this, when the cell membrane gets separated from the corner, this is second, that is incipient plasmolysis. Okay, but in the first limiting plasmolysis, what will happen? The turgor pressure becomes 0 here. How? See, understand it once again, you will get clear it. Okay, so first in limiting plasmolysis, what happens? The plasma membrane is there and cell wall is there. Initially, when it is a turgid cell, the plasma membrane is pressing the cell wall. But here it stop applying the pressure. So in limiting plasmolysis, cell membrane or protoplast stop exerting pressure on cell wall. And when the cell membrane or protoplast stop exerting a pressure on the cell wall, the turgor pressure becomes 0, and that is a limiting plasmolysis. When more water loss continues, the time will come where the particular cell membrane gets separated from the corners, and that is incipient plasmolysis. And if this continues, the more water loss will occur, and in that the particular protoplast will come inside a one particular corner. So this is a cell wall, and this is a cell membrane. And such a way the cell membrane gets separated from the corner in this direction, and this is the space. Now this event is known as evident plasmolysis, that is the third step. So in first step the turgor pressure becomes 0, here turgor pressure become negative, and here the turgor pressure become more negative, and this is a plasmolyzed cell. And when this is a plasmolyzed cell, the turgor pressure become more negative, okay, in that. And here the outer solution, because cell wall is permeable, in the outer solution here you will see the presence of hypertonic solution. Such a way the stepwise the plasmolysis will occur, and this is a plasmolyzed cell. Now after, if plasmolysis continue in these three steps, initially this phenomena is indicated by a shrinkage of protoplast leading to the separation of plasma membrane from the cell wall at the corners, that is what we can say the incipient plasmolysis. And after that the plasmolyzed cell is obtained in the evident plasmolysis. So when the cell is left in the hypotonic solution for more time, the protoplasm completely shrink. Now the cell is said to be plasmolyzed, and the movement of water occur across the membrane, moving from an area of higher water potential from the cell to the area of lower water potential, that is the outside of the cell. Thus, normal living cell when kept in hypotonic solution thus becomes placid because it allows the entry of water as well as the outward movement of water takes place. So it is become a flaccid. In a flaccid cell, the pressure potential become zero. Hence, the water potential become equal to the solid potential, and that we will discuss. Plasmolysis does not normally occur in a nature, with the possible exceptions of extreme water stress in the saline environment. The salting of pickle and preservation now plasmolysis is helpful also. It has a beneficial role as well. That salting of pickles and preserving of fish and meat in a salt are good example of practical application of plasmolysis. When we salt in them, what will happen? Whatever the bacteria will come and or fungus will come and sit on this particular salted pickles or the salted components, they will plasmolyze this particular animal cell get plasmolized. That is a bacterial cell or fungal cell get plasmolized. So the infection will not occur and they will not harm this or degrade these components. So when the cell or tissue is placed in isotonic solution, there is a no net movement of flow towards inside and outside, but flow can occur inward and outward, but net flow movement is zero. So the external solution balances the osmotic pressure of the cytoplasm. So it is said to be isotonic condition, and the both are in a state of equilibrium. The net movement is zero. The process of plasmolysis is usually reversible. The process of plasmolysis is usually reversible, and when the cells are placed in a hypotonic solution, water diffuses into the cell and causing the cytoplasm to build up a pressure against the cell. The cells are placed in a hypotonic solution and water diffuses into the cell and causing the cytoplasm to build up a pressure against the cell wall. And that is the what when the build up pressure against the cell wall. Okay, then this is called a turgor pressure and it is known as turgid cell. So pressure exerted by the protoplast due to the entry of water against the reservoir wall, it is known as pressure potential. So turgor pressure or pressure potential is the same, and turgor pressure or pressure potential are the same when the entry of water takes place, and due to entry of water, when the particularly we can say the cell becomes swollen, and when the protoplast exert a pressure on the cell wall in a hypotonic condition, that is known as turgor pressure, and the turgid cell are obtained. So because of the rigidity of the cell does not rupture in case of plant cell in the hypotonic condition, and the turgid cells are obtained. Turgor pressure is ultimately responsible for the enlargement and extension of the growth of the cells. Okay. So now we have to understand turgor pressure in more detail because plasmolysis we have discussed. So if we talk about turgor pressure, it is a pressure exerted by cell wall. It is a pressure exerted by a protoplast against cell wall. So when a pressure protoplast exert on a cell wall, it is known as turgor pressure, and the cell will be turgid. So suppose this is a cell, and this is the plasma membrane, and this is cell wall, and this is a plasma membrane. So when a water enters, the plasma membrane or protoplast will exert a pressure that is known as turgor pressure, and opposite, the wall will also exert pressure that is known as wall pressure. So the pressure exerted by wall pressure exerted by cell wall against turgid protoplast is called turgor pressure. So when the pressure is exerted by a cell wall against the turgid protoplast, the cell is known as against the swollen protoplast, then it is known as a turgor pressure. So turgor pressure, according to Newton's third law, the pressure is equal and opposite. So turgor pressure is equal to wall pressure. Okay, so in flaccid cell and turgid cell, In turgid cell, turgor pressure is highest, or we can say positive, but in flaccid cell, turgor pressure is equal to 0, and in plasmolized cell, turgor pressure become negative. Okay, so these are the three things are seen in case of the, we can say, the turgor pressure. This turgor pressure is not applicable in a free solution, that is a solution in a vessel. This is only applicable for osmotic system, that is system separated by a semi-permeable membrane. And obviously this turgor pressure helps to maintain the shape of the cell because it is responsible for the enlargement and extension of the growth of the cells. Okay, so this is what a turgor pressure, and that is all about the plasmolysis. Now after plasmolysis, one more phenomena we are going to discuss here, that is imbibition. Okay, and when we talk about imbibition, it is also a special type of diffusion. So it is a special type of diffusion where water is absorbed by the solid colloids and causing them an enormously increase in volume. And if you take example, the absorption of water by the seeds and the dry roots. If we take example of the solids or hydrophilic colloids, then the examples are the seed and dry wood, and they absorb this water and they increase in size. So this is a wood. This wood gets swollen when placed in the water, and increase in volume occur. Sometimes you will see this is a what a shrunken fruits are there. When they are kept in the water, they get swollen like this, and this is due to the what imbibitions. Okay, so the pressure that is produced by the swelling of a wood was used initially by a prehistoric man to split the rock and boulders. It was used initially by the prehistoric man to split. So if two rock are there and we want to separate this, what we will do? We will put a piece of wood and allow them, pour them with the water, a plenty of water, that will swell up. Now different type of organic substances have different imbibing capacity. Okay, that we will discuss, but before that we have to understand imbibition in more detail. So here it is obviously a special type of diffusion. Now what will happen in case of imbibition, that we have to understand. So see, suppose this is a wood, and this wood has this solid colloid pieces. Now what will happen in this imbibition? The water get absorbed, and this water will reach the intercellular spaces or between the spaces, and the swelling of wood will occur like this. And that is obviously due to the accumulation of water between the cells. So this here, this is a solid which is absorbing solid colloid, which is absorbing the water, and that is known as imbibant. And here this imbibant get swollen due to entry of water. So the water which is being absorbed, that is known as imbibate. So imbibate is a water and imbibant are the get swollen. So imbibant can be what? Seeds and dry wood. They are what? Imbibant. They enormously increase. Volume of this get enormously increase. Now, what is the simple prerequisite for success of imbibition? So it is a adsorption. Now what will happen here? We remember one more thing. If this is the solid component, what will happen? On this solid colloid, the water molecule will adhere. They get absorbed. without dissolving this solid component. So here we should remember that the water or solvent get adsorbed over solid cells without dissolving them. Okay, so that is the what thing occur here. Now here, when they adhere or adsorb, the what the heat is released. So heat is released during the adsorption as well. So what are going to occur? So what are the observes or what are the result of imbibition? First is volume of imbibant increases enormously. Volume of imbibant increases enormously. Second, imbibition pressure, which is also known as metric potential, that develops. And third, if we talk about the third, then the heat is released during adsorption, and this heat is known as heat of wetting, because it is an exothermic reaction. Okay, it is an exothermic reaction. So without dissolving a solid component, when the volume increases due to the adsorption of molecules over the solid component, then it is known as a what imbibition. So if we talk about one more example of the imbibition, during monsoon season, I hope you have seen that our doors do not close properly. Why? Because of the swelling. But what? Because the moisture is more in the surrounding due to their absorption occur by these dry woods which are present in the door, and swelling of door occur due to this the proper closing that cannot occur. Now, what is prerequisite? Now, what is prerequisite for success of imbibition? First, the requirement is water potential gradient. Means water potential must be there. Gradient means that one should have a high water potential and one should have a low water potential. So water potential gradient between absorbent and liquid imbibed, that is between imbibant and imbibate, is essential. And second is there must be a affinity between adsorbent or imbibant and imbibate. Now affinity means the imbibant or a dry wood should be hydrophilic to absorb the water. If it is hydrophobic, it won't be absorbing water or absorbing water. Imbibition cannot occur. Okay, so affinity should be there, and obviously dry woods and dry seeds have affinity. Now, in application, we have discussed during the prehistoric man The swelling of wood has been used by prehistoric men to split rock and boulders. Seedling emerge or seed germination occur due to imbibition. And now when we talk about some more facts of imbibition, the power of imbibition is variable in different substances. Or imbibition imbibition pressure vary among different substances, and in that the imbibition pressure of agar agar is greater than pectin, and it is greater than proteins, greater than starch, and greater than cellulose. Okay, so agar agar has the highest imbibition pressure. So proteins have a very high imbibing capacity compared to the carbohydrate, obvious. Because of this, we have written protein have a greater than imbibition pressure than the starch. And that's why proteinaceous pea seeds swell more on imbibition than the starchy wheat seeds. Okay, so this is the reason. Okay, so that is what about the imbibition. Some MCQs are there. When the external solution is more dilute than the cytoplasm, the solution is said to be hypotonic. Obviously, that is a dilute solution. Cell in hypotonic solution, that is going to swell, that is a turgid cell. Plasma is occur in hypertonic solution. Okay, so that is what about the particularly we can say the plasmolysis and the imbibition. So this is all about what we can say the osmosis, plasmolysis, and imbibition. Three mechanisms. Now two more mechanisms of water transport are remaining. Water plant water relation are remaining, and that is DPD, diffusion pressure deficit, and the water potential. That we will discuss in the separate lecture. Okay, I hope you understood these three topics. Thank you everyone. Thanks a lot.